Welcome everyone to the January edition of the Beta Break. This edition is organized in collaboration with the Women in the Infective Science student chapter of the University of Amsterdam, short with students, and SPO 25. I'm Marwa, I'm here with Rosie. We will be your moderators today. Off screen, we also have Frank, who is moderating the questions of the audience. So if you have any questions, please leave them in the Q&A. Frank will select your questions and we will try to answer some of these after every section. For those that are not familiar with the Beta Break, we are a student-initiated science discussion platform of the University of Amsterdam Faculty of Science. We host monthly panel discussions on a broad range of subjects within the natural sciences. Rosie.
And as Marwa told you, I'm Rosie and I'm part of the WIF Students, which is an organization that serves the interest of female STEM students at the University of Amsterdam. And today we will be talking about a revolutionary gene editing technique, CRISPR-Cas9, for which the microbiologist Emanuelle Charpentier and the chemist Jennifer Doudna won the Nobel Prize. And our speakers can tell you a lot more about this later, but first I will line out the program for you today. So first, the speakers will give a short introduction about themselves. Afterwards, we will be discussing the technology itself. What is it? Followed by the ethical questions surrounding it, and finally, the role of women in STEM. And you can leave the questions in the chat throughout the whole event, and we will try to answer them after every section. And otherwise, we also have some time at the end of the discussion to answer some more questions. So for now, I would like to give the word to Wen to introduce herself, followed by Pernette, and last but not least, Jantien.
Yes, good evening, everyone. I am Wen, and I'm currently a PhD student at the Laboratory of Microbiology. I work under John Vanderoos, who is actually one of the pioneers in the CRISPR-Cas field. And what I study for the past few years is I study the fundamentals of CRISPR-Cas systems as bacterial immune systems, but also the application of new CRISPR-Cas proteins. So it's not just Cas9. Yes.
Thank you, Wen. Pernette, would you like to introduce yourself?
Yes. So I'm Pernette Verschuren. I'm associate professor at the University of Amsterdam, where I'm leading a group on functional organization of the genome. So we are actually mostly interested in the epigenetic regulation of the genome. So this is a level basically on top of the DNA, driven by all kinds of proteins and driving the organization of the chromatin. Now we are interested in the causal relationships of various proteins. So therefore we actually use engineering tools. And we're mostly interested to understand this really combining experimental tools and also computational tools, so multidisciplinary angles basically, and to apply this because we are interested not in the stability, because epigenetics basically drives cell type identity, and this is considered to be very stable epigenetics, but there is also a lot of plasticity and dynamics. So we are very much interested in this variability and how cells are actually really changing in time. Now, last year I've collaborated and coordinated a huge European network where we focused on epigenetic gene regulation in breast cancer resistance development. So also understanding how this actually leads to maybe pathological changes. So for now.
Yes, super interesting. Jantien, would you like to introduce yourself? You're on mute.
Thank you, Marwa and Rosie and SPO 25. It's great to be here. I'm in Boston at the moment. I'm a philosopher and ethicist by training. I work since many years on the work floor of the lab. What you see behind me is, of course, a virtual background, but that it is the lab where I work at the Reeds Institute for Biologically Inspired Engineering, which is not only bioengineering, it's biologically inspired engineering, and it can also be like mechanical engineering, material science engineering, and so on. But the inspiration is from mechanisms in nature. That is, of course, also for a philosopher, very interesting. I work primarily for the organ-on-chips platform, as it is called, and the synthetic biology group. And the synthetic biology group is led by George Church. I've been working since around 2006 with George Church, and I was more or less an eyewitness to the early investigator meetings on genome editing and using CRISPR for genome editing, which was in retrospect very exciting. I'm also an assistant professor at the University of Groningen, and my affiliation there is with ERIBA, which is the European Research Institute for the Biology of Aging, that has also a dedicated CRISPR facility. So that is my Dutch connection. And by the way, I studied philosophy at the University of Amsterdam, and I got my degree from there. So it's very great to be home here with you all, and thanks again.
Thank you, and thank you for being here. We are really glad to have you three as panelists. The first question, we expect a broad audience, so it's important that everyone knows exactly what CRISPR-Cas is. I would like to ask you, Wen, to explain to us what is CRISPR-Cas.
Yeah, so I think a lot of people, so okay, how would I always prescribe this CRISPR-Cas is a coin, and there's two faces of the coin. There's one part of the coin that is the fundamental part where CRISPR-Cas came from, and then there's the other side that people mostly know about, and that is CRISPR-Cas technology. So first on the fundamental part, so CRISPR-Cas systems, or CRISPR-Cas is a immune system found in a bacteria. And what's really beautiful about this system is that is an adaptive immune system. So there's like a memory system within the CRISPR-Cas defense system. It's a immune system against viruses, just like humans have viruses, which is one that is running around now. Bacteria also have viruses all the time. So you can imagine like one bacterial cell is a factory that's doing its thing, and then all of a sudden you get invaded by viruses. And you can see CRISPR-Cas system as kind of like the security group within the cell. You have the parts of CRISPR-Cas, like I would call them the geeks, and they look for information, and they get the immunity, and they put it in their database. And then you have like the big tough guy, the bouncer, who uses information from this database to then look for like, oh, okay, this is the picture of the virus, and then we will like cut the virus. And what people have found is like, you can change the information of which you give the paper to the bouncer. Instead of the face of the virus, you can say, oh, don't find the virus, find something else, like find this sequence. And this is how, and what people did was they took the bouncer guy from the cell, the bacterial cell, and they put it in another factory, in a human cell. And they tell the bouncer, go find this piece of DNA and go cut it. And that's basically the CRISPR-Cas technology and how it started. So how you can see Cas9 is the bouncer. It is a protein that finds DNA and cuts it. But now what people have done is that sometimes they remove this cutting ability of this DNA. So it just finds DNA and just binds it. So you can see it hugs the DNA very well. So I guess then I would say that kind of goes to Pernet, what Pernet is doing right now, which is also very exciting.
Thank you, and that was really funny how you explained it, but it made it really clear. Pernet, you're using another kind of Cas9, one that doesn't cut. Can you tell us more about that?
Yeah, so we are using the CRISPR system. We're also using it to actually integrate pieces in the DNA or cut the DNA, make mutations. But mostly we use it actually to understand epigenetic gene regulation. So there we use it in a different way. So we use the Cas protein that is mutated. So it's not any longer acting as a cutter. So we use it as sort of a targeter. So we still use the guide. So the guide is able to bind specifically to genes that we want to bind, that we want to actually modulate. But to the Cas, we bind epigenetic regulatory proteins or enzymes that are known to give an epigenetic effect. And this way we can specifically target the specific gene that is targeted by the guide. And that is then recognized by the Cas protein. And then the epigenetic effect is actually established there where the guide is binding. So this way we can make a certain epigenetic modification, and then we can test actually whether our hypothesis fits and whether indeed what we thought or what we predicted is true. So that is what we're doing. So that's really interesting, because also under the CRISPR system, there's of course all the epigenetic regulation. And this is also something that is basically still difficult, because we don't know everything of the epigenome. As I said, we are interested in the dynamics and in the variability. And this is something that is not yet developed so much, the epigenetic regulation. So that's why sometimes the system is still sort of unpredictable. So we're very interested in this. And I think that this specific form of epigenetic targeting, so we call it epigenetic editing, is also interesting because you do not make actually a mutation in the DNA. You do not make a genetic cut. So you leave the DNA actually unharmed. So you're also not activating the DNA repair pathway. So maybe in the sense of, if you would like to use it as a co-therapy or treatment, then this would be more mild, because you're not changing the DNA, but you're changing the epigenetic layer. So you're changing actually the regulation of a gene, but not per se the genetic code.
Thank you. So if I understand correctly, like originally CRISPR-Cas, well, it comes from bacteria and we can use it to cut and pass new DNA pieces. But what you're doing is you don't necessarily cut it, but you edit like the epigenetics. But in both cases, I would say,
so the system consists of two parts, basically, in the engineering form. So you have the guide protein and you have the Cas. The guide protein binds very specifically to something where you actually want it to bind. And the Cas protein is originally the nuclease, so it makes the cut. Now we have a mutated Cas, so it doesn't cut anymore. We make a protein, a fusion protein. So we fuse the Cas to an epigenetic regulatory protein. And then this actually works the same. And it just goes to the guide, and there it gives its effect. So now the epigenetic regulatory protein is doing its job on the guide. So you have an effect, but you're not making a cut. So making a mutation or then integrating another piece of DNA. So it's a bit different, but it's still the same way of engineering, basically.
And whether you are cutting or whether you are editing, like the way you are doing, in both cases, like you're changing one gene, but that gene is part of a whole network. And since we are changing one piece of a network, you probably are shifting something in the whole network. So do we really know what exactly is happening? Aren't there a lot of side effects if we do this? Maybe you or Wen can answer this.
You want to go first, or should I? Yeah, I mean, okay, so maybe I'll start, and you can always add on, Pranat. So I think, like, look, there's many networks, and people, it's not like people study diseases in very small cells, and everything is very regulated, or everything is tested before we ever go into real human applications. And per se, and I think right now the genetic diseases that people are normally tackling on a DNA level, I would say, are very small, little mutations that are missing that is repairing what the mutation caused. For example, if I have a muscle disease, I think I have one mutation here, and you're just repairing to what it is originally. Again, we are not in no means to the part that we are messing around that, oh, how can I get more muscles, and how that network kind of works. I would say, I don't think in disease curing or gene therapy wise, I don't necessarily think we're there yet for that. Maybe Bernat can add on.
Yeah, I think it's interesting because of course we always think that we can actually, that there is one gene underlying a disease, but that's of course a really naive idea. And it's a whole network of genes that are in balance, that are communicating with each other. And then one gene is producing a factor that again binds on the other. So there is feedback regulation. So there is huge complexity of the network. And yeah, if you change one gene, you can indeed consider that the whole network starts to actually breathe in this whole action. So yeah, I think that for now it's difficult to predict everything of this. And of course, there are huge genome-wide screens that try to understand this and what is going on. But there is still also challenges because, as I said, the genome and the epigenome is very flexible and actually quite dynamic, so that you need single cell analysis, you need temporal analysis to really understand what's going on at the fundamental level. There is still many things to solve, but yeah, that's also still the interesting part.
Thank you. And we actually immediately have a question from the audience. Someone is asking, do you think that CRISPR-Cas can cure cancer? I would say no, but I would love to hear your opinions. Ben?
I think so. Cancer, I think we kind of go into the direction of CRISPR-Cas being used as kind of personalized medicine, because that's like cancer is not, I have this cancer, you have this cancer, we can edit the same gene and it will be fine. It's, I have several accumulated mutation, thereby leading to cancer, and you have several other accumulated mutation. I think, yeah, you can, cure is a big word, but maybe you can prevent cancer, I would say, in the future, because again, cancer is based on accumulated mutation that leads to why it is proliferating, I would say. So I do see it as a possible personalized cure for that in the future, not now.
Yeah, I also think in this way, because it is really complex. And of course, cancer is not one thing. So there are many different cancers. And for some sort of immune therapies, people are actually changing the immune cells out of the body and then replacing. And that's, of course, much more easy than doing something within the body. So, but of course, it's an engineering tool that leads to many different things. So if it's really widely applicable, then yeah, it has enormous impact. But there are still many challenges, actually, how to get it into the cells, how to specifically target, are there off-target effects? So I think before we end in real treatment, we're still, in the beginning.
And then something totally different, because the Nobel Prize for CRISPR-Cas is one in the category, is one for chemistry instead of medicine. And I was curious, and I guess a few other people also, why chemistry instead of medicine? Pernette, do you have an answer on that?
Yeah, I think because it's, of course, it is an engineering tool, which actually stems from chemical design. So thinking about what kind of chemical design is underlying the regulation of bacteria in their sort of immune response. So basically, it stems from chemical technologies and using it to engineer and to change. What maybe also helps is that the Nobel Prize winners are from the chemistry field, from the microbiology field. So they also come from the more fundamental field than from the medical field. Now, of course, the technology can have huge impact, and then it could easily be also a medical Nobel Prize. But for now, this is still difficult. So I think it is, for now, really the tool that is developed and all the knowledge with it, how this works. And it's, of course, a really interesting development and idea. So, yeah.
Can I add on quickly? So it's exactly what Bernard said. It's an engineering tool. I just want to say, I want to stress like how great this tool has been, not only for the medical field, but also for any research in general. Never had it been so fast that someone can create precise edit in plants, in bacteria, and with such ease, I would say. And that's the the power of this tool, and thereby chemistry. Yes,
that's a really revolutionary tool. So Jantien, you're also there from the beginning of this revolution. What do you think this technology is Nobel Prize worthy besides these things, and why now?
Yeah, I really want to emphasize that I completely agree with was just when, and Pranette mentioned before, that CRISPR technologies, and CRISPR-Cas9 being one of the many CRISPRs even, but there are also other, even totally non-CRISPR genome editing technologies, so it's important, but it's all tools. And I think if you would ask, like, so these tools, where are they, say, in the numbers? Where are the most, say, CRISPR activities carried out? For what purpose and what place? And that is probably just like a wild guess. I think that for 95% of the CRISPR users are at the benches of fundamental labs, because indeed it is used to, it's an enabling technology, and it's very, very basic. And a lot of the work done with CRISPR systems is not at all exciting for anyone outside of the field, but it has opened up the avenue for that work, where the fundamental science is, of course, I would say, at the basis of anything like therapies and, like curing cancer. That would be great. I think we can all agree on that. But like, even if we make a little progress, that would also be great. And as we see it now, like if you would go for genetic therapies or gene therapies, then CRISPR is probably indeed one of the necessary tools nowadays. So it's really this instrumental use, and such an important instrument. And in that sense, it's really revolutionary, the move from this being a natural tool, or a tool owned by bacteria and only used by them, moving that to the lab benches as a tool for scientists, for engineers, biologists, chemists. I think that is that's right. That is worth of a Nobel Prize. I would say so. It's of course a personal view, but I would really I was very happy to see it, and I think it is absolutely well deserved.
Yes, very nice. So I hear you say that it has a very broad application, and that's also why it should be Nobel Prize worthy. I also heard you say in the beginning that it's a quite simple technique, and this makes me think about an episode of Tegenlicht, which I saw lately from the Dutch channel VPRO, and they made it seem like that every student or even a child would be able to apply this technique. And there are even biohackers at this moment who are selling CRISPR kits to the general public, including all the necessary tools to carry out such an experiment. So when you're in the fundamentals, I would like to ask you, how realistic do you think it is for non-experts to do gene editing in organisms?
Okay, first let me say this man gives me a lot of stress, and his work as a biohacker. I don't think. Okay, let's go through several parts. He sells a kit where you can inject yourself, and there's editing. First, do you know for sure you have this sequence that the Cas9 is looking for? Second, delivery is one of the biggest bottlenecks in CRISPR-Cas delivery, and we are still working towards efficient delivery, or the people are still working towards efficient delivery of CRISPR-Cas. If you're gonna inject it into one cell, I don't know. I just don't think that the efficiency, or even if you have a protein, you have a protein already with your guide, and you put it in your one cell, the protein might have been degraded after a while. And how do you would never screen, like for example, even in the lab, when people put the DNA expressing Cas9 or protein expressing Cas9, we're talking about low percentage. We're never talking about 100% delivery. And then of that, then they screen which one has the edit. And I just don't think it's that easy, because there's not thousands of researchers doing research on how to do this in a gene therapy in a enclosed lab, of which can lead to someone selling you a little kit that say, please inject this in your muscle. So in short, no, you cannot do this. But maybe Jantine and Bernard have other things to add, or a different opinion on it.
Yeah, I also agree that I think this is very wild, that, it has to go into the cell. You have to, if you would inject, then you would propose it would immediately go into a cell. I mean, that is, of course, yeah, completely, as Wen says, that this is a bit overestimated. But of course, it is very difficult. And I think also it is very dangerous if people are starting to do these kind of things, because you've no clue what they are doing, what they are making. So I think these hackers are really dangerous and they should be stopped, because I don't think it's a good thing. They have no idea what they are doing. They're messing up with stuff. So, yeah.
Yeah, those are two very, very clear answers that it shouldn't happen and shouldn't be legal. So thank you for that. And we already know, at least. Then we continue with some questions from the audience. So the first one is, what is the error rate of this technique?
Yeah, so maybe, so the beginning of the Cas9, so what they mean with error rate is, for example, it has to find this 20 letters to cut it, but then it finds 18 letters that matches the 20, but one doesn't match, and then it still cuts it. So if you think about 20 letters and you think the human genome is, I guess, 3.1 billion something letters, there is an error rate, but people have done like a lot of research on this. And what people have done is they have re-engineered the Cas9 to be now a lot more precise, and they're still looking at these precision-wise. There are new variants of Cas proteins that are more precise as well. So I think the error rate is a lot lower, and I think it can be very, very precise at this point. But then again, I think for every sequence that you target, if you end up doing it for gene therapy, you have to always double check where else it can possibly cut and where else can it cut.
Okay, thank you.
Yeah, I also think so. Yeah, of course. So when we started with the CRISPR-Cas9 system, so with the epigenetic targeting system, there are plenty of studies that show that, of course, there is the low-hanging fruit, and they publish the really nice and interesting things. But also, I hear many stories from researchers that start targeting the guides and then looking for epigenetic effects, and then there is still many things that are unknown and that we don't know exactly how the epigenome or the genome is regulating, how other regulatory regions are affecting the effect, how actually you really get the effect. So to systematically get the effect to any gene, I think we're not yet there. So I realized this. So now with the consortium of Dutch researchers, we're applying for a big grant to actually sort out whether we can make this more effective, whether we can understand the guidelines of underlying epigenetics and underlying this targeting, that we can actually sort of, try to find a certain set of rules for specific genes, how they are regulated and organized in their specific context, and then what kind of tools would you use for those classes or other classes, to make it really efficient.
Yeah, no, I see. Yeah, somebody else also has a question about CRISPR-Cas9, because this person says that it's already used in patients. And do you think that this technique of interfering in epigenetics, that it interferes like CRISPR-Cas9 with the CRISPR-Cas0 technique?
I'm not 100% sure.
No, the question was, is it used in patients? But based on your answers from before, I would say no.
We are not yet that far that things are being used in patients. So I don't exactly understand the question so far, because I think at the moment we're using epigenetic editing for finding out causal effects and screening. And of course, the hope is that we can use it as co-targeting of monitoring, or in the end, indeed, to treat patients. But I think we're not that far.
Thank you. And well, you can already hear it from the questions that are asked, like what kind of side effects are there in patients? And these kind of questions also bring some ethical questions in mind, because originally CRISPR-Cas was made, was further developed to understand the immune system in bacteria and to use for agriculture and maybe to cure genetic diseases, but it can be used in a really broad way. And a lot of people, when they think about gene editing, they think, for example, of designer babies or women, people with tails or wings. And these kind of things really raise some ethical questions, which we want to discuss now. One of the first questions I want to ask, actually all three of you, is where should we draw the line? Like, for what should we use CRISPR-Cas and for what shouldn't we use CRISPR-Cas? And maybe as the bioethicist, Jantine, want to start?
Oh, you start with the most difficult question, because it is not possible, it's not reasonably possible to draw a line. Because what I mentioned before, this is such a, say, general tool, and there is so much that also Parnett and Ren said, there is so much that we still do not know, that it would be completely speculative to draw a line. The drawing that line would be so general that it simply, it doesn't work. Like if you draw a line, like say, it should never harm people. Yeah, that's like the same. That's like the same. The general, say, medical ethics rule. First, do no harm, but that does not tell you as a doctor at that moment with your patient what you should do. So I'm very wary personally of making such statements. It also might give people like a false reassurance, like, oh, okay, yeah, so yeah, now we know, where to go and where not to go. And that is not how science works. It's one of the interesting things for me, being since so long working with the scientists in the lab, being in all their meetings, I hear firsthand how difficult things are, how unexpected the course is that things take. And yeah, like, curing diseases, that's, for example, and at what level. You can have it as a far aim, and I think we later speak about this, like the baby thing. That's I'm very I think that is it's sort of like fantasy land, and we should. It is very regrettable. I personally, let me say it this way. There may be applications in humans. I think it's very regrettable that at this moment, so much of the attention, and like CRISPR is immediately linked to whatever things with babies, which I said to you before, most of it is used to tinker in tiny microorganisms at the bench. So much attention goes to these sensational, speculative applications. Also, a lot of money goes there in ethics committees, name it. They're all talking about preventing precisely this. And that is like a wrong way of paying attention to this technology. And ultimately, what people want to prevent is, say, rogue use, like for really evil purposes. Well, that you will not stop by pumping a lot of money and effort and personal expertise and resources in something that is really, that's not reality. So that's actually my greatest concern. I do not focus on as an ethicist on the perfect baby thing. Of course, everywhere where I show up, or students come, or school high school kids send me emails like, oh, we are writing a project, whatever, and we want to talk with you about perfect babies. And that is the only thing I can tell them, is that I do not think that's the most important ethical topic that we need to address. And it's actually a distraction from what is really important.
I want to respond on that, but I first want to hear Wen's and Pernette's opinion, where to draw a line.
I think Gentine had a very good answer on there's no line to draw. No, but I think, like, at least on my side, I think I definitely agree with the whole designer baby. It's the technology is not there to kind of go back to the babies. Okay, designer baby. You cannot see how tall I am. I am a very short person. I would love to be taller, trust me. But to become tall, the genes, the network, we talked about that. There's so many things involved to just making me maybe 10 centimeters taller. The research, the fundamental research is not even there. How dare we even think that we are gonna modify a baby that will become a tall baby or tall person tomorrow? There's no way that's happening. It's really not. We just don't know enough yet on genetics wise. But what we have right now is a technology tool that I would say that can do a lot of good. For example, modification in plants, gene editing plants. If you think about it right now, like people obtain plant strains by very different others technique. One of it is just putting UV waves and damaging all the different DNA, and then you get the phenotype, the plant that you want that has, I don't know, thousands, 10,000s of modifications, but you got the plant that you want. But when it's about CRISPR, you cannot modify or take out one gene that is super precise and your plant stays the same. To have, apparently, especially now, climate change is changing. There are droughts, plants are having a tough time growing. I think now is the quickest way to create new plant strains, to have these plants be able to grow in very, very harsh conditions is the quickest way we get there. The current technology that we have right now is slow and it's very, very expensive. And I would say definitely on the plant application, that is really that it should go. And I think on research-wise, that already is ongoing, and I think people should keep it up. If in the future we actually reach the point that we can cure genetic diseases, that will be amazing. However, I talking about design baby, of course, for me ethically that is not okay. But I also don't think we will reach there because the science is not there, really not there yet. So Pernette.
Yeah, I think that's, I would say the same thing also. And that we're completely not there yet because we do not understand the complexity of many things. So the idea that we could change one gene and then, yeah, design a baby is at this moment, yeah, I would say off scope. And yeah, but thinking about diseases, of course, if you could make a change, that would be really interesting. And sometimes I also think, well, you can also think it the other way around, and you could say it's actually unethical if the method would be there to not allow people with a genetic disease or, you know, to be cured. Or would you then say, okay, you're not able to get it, we can do it, but yeah. So it's always very difficult, I think. And yeah, I also completely agree with the plant research that there are, this way would be much more elegant than throwing chemicals or mutations with UV, or yeah, how you now make, actually you select is also a way where you do the same thing but on another way. So it's always a bit challenging, also considering yeah, Louise Brown, sort of the in vitro fertilization. I mean, if the technology is there and the people want it because they cannot get babies, then, you know, they need it and it will get there. But now it's such an over, you know, exaggerated thing that now that this engineering technology is out there, that we could use it for dangerous things. Yeah, of course, we need to consider and have societal discussions. what we can do with this technology. But there are actually also other technologies that can do exactly the same. There are zinc fingers and talents. There are other systems that exist longer. So there are big companies also making those. So yeah, it's always a bit over the top that when there is this really interesting tool that people immediately start having these ideas about design babies and how far can we get and are we going to make a different world? Well, maybe we make a different world, but that's also how we are. If we maybe evolve into something using chips or maybe integrating, whatever technologies in our body, I mean, maybe that will come. And then maybe also at some point we can use the CRISPR for some elegant ways. So I think also, people make things and engineer, and then technology grows. So we just, we evolve in the things we can do. So, yeah.
Well, the main argument all three of you gave is actually we are not there yet. But I can imagine that maybe in 20 years we will be more there. So like at the beginning of this century, we had the Human Genome Project. Before we didn't know what the sequence exactly was. Now we know the sequence of the Human Genome Project. And I can imagine that in the coming years that we will know more about the epigenetics, about the network, how all these genes communicate with each other, that at a certain point we will be there. So shouldn't you already have these discussions? You already said it, Bernard, but it still seems like you all think like, oh, it's not important now. But I think, you can also say like, okay, we can use it for example, the plants and for curing genetic diseases, but we won't use it for things that are not necessary. For example, changing your hair color or eye color. You can also draw that line, or do you think that we shouldn't do that?
Yeah, I think that's also a very difficult discussion if you start to think about it and from a sort of philosophy point of view. So maybe for Jantien also to think about, because if you think about, okay, you want to use it for something that is good, so you want to use it for something that is good, or you do not want to use it if it's bad, then what is good and what is bad? So it starts being very difficult, actually. You get in all kind of discussions of what is good and what is bad and what is still wanted. And I think that gets very difficult. And of course, it's interesting to have those discussions, but also it shouldn't hamper the progression of interesting technologies and the application of it. But maybe Jantien has also something to say about this. It's interesting stuff.
I agree with you, Bernard, and maybe to put things in a time perspective, to give one example. So one of the applications that is very near of genetic therapy where genome editing is used is finally gene therapy for sickle cell disease. I think in the US, a patient has been treated, but I'm not sure. I'm not sure at this moment what a genetic intervention was. But then you must think back and realize that in the mid of the 80s, the first disease that came, there were two diseases that popped up in discussions where gene therapy would be great. And there were the immunodeficiencies, the inborn immunodeficiencies on the one hand, these bubble boy children, and the hemoglobinopathies, the sickle cell disease on the other hand, because they thought that this is at least genetically simple. So, and they had an idea of how to do it. Of course, we know now in hindsight, as you mentioned, only in the early 2000s, we knew the whole human genome sequence in the 80s, as they have done it. They have tried it. They have tried to with a very clever plan, and it seemed logical and plausible and ready to put in humans. They have tried to make a gene therapy, do a genetic intervention for immune deficiencies. Well, that went terribly wrong because a number of those children, so they put indeed out of the body changed, say, genetic constructs into those children back again. But as we didn't know anything about genome, they basically, as we must admit now, they didn't know what they were doing. And what happened was that, yeah, there was a change in the genome of these children, but unfortunately it was in an oncogene next to the immune disease gene. And these children then, a large number of them got leukemia, and many of them did not survive that. So I think that should be, it's always good to look back in the past and to, you know, to learn from those lessons, because this seemed absolutely plausible. The patients and the parents, of course, wanted this. And the doctors were and the scientists were convinced that this would work. This was the logical and correct way. And it just went terribly wrong. And then, of course, it was sort of the out of view for quite a long time. Even now, we are not yet at the point with having CRISPR now, which is like sort of a mega change compared to those years. Even now, we are not able to simply make that construct, put it in an IV infusion, and give it to a patient, and it's going to work, because that is not appears not to be the way it works. And I think that also from those lessons, there should be, say, on the technical side, the realism and the humility to say, like, when we talk about things like embryos, and then it should be heritable, the next generation should have it. Well, when would you see that? Well, that would like take like 18 years or something. Like the feeling that we could do that. And I think that puts a bit in perspective the urgency of discussing the do's and don'ts in it. And probably, as what Wen pointed out, the agricultural use, like making drought-resistant plants. Well, you could say that would happen in nature by evolution over a very, very long time. If the climate demands it, plants would adapt. Very long time. And at this moment in our world, we cannot wait for it. We do not have the time. We do need to use the technological tools that we have. So I think it's of course a personal view, but I feel more a necessity and a moral duty to discuss, say, those uses instead of speculations about, well, what would then a perfect baby or a super baby be, or so. That it's so remote from saying, here is a drought, climate has changed, and we see it's exponentially worsening, and what are we going to do now? And that's also what the world, by the way, is asking us, is asking the scientists, like, do something. So that's also why I work in the lab, because I feel that, say, also as a moral duty to promote those uses of technology, rather than, well, sit in a circle with whatever philosophers or lay people and discuss about what degree of perfectness in a child would be acceptable or not. So that's like how I see it. And with the ecological application, there are a lot of problems there too, and they're very complicated. So where to invest our efforts? Well, I think it should be, for me it is there. And yeah, treatments for diseases that are realistically within reach, and they will probably be in, say, born people, and not say in unborn children or early embryonal or gamete stages. That's really very, very remote.
Yeah, so can I add something? So let's just say, okay, 50 years, 100 years, 1,000 years, we get there. Okay. But then again, I ask you, then you say, okay, we select for, I don't know, superhumans. Let's say that in this parallel world that does happen. IVF, medicine, right now, nowadays, this is selection. People, you screen for embryos and you decide which one to keep. And those are selections that are currently already happening, and then you shun a technology of which can bring so much good. I would say, okay, so let's go back. Surgery, right? Yeah. So I think in the hospital, many people do surgery every day. You have surgery that saves lives, but at the next hall, you have surgery that makes breast implants and liposuction, and those are cosmetics. And I would say, yes, those are two sides of the same technology, like surgery, and this is the case. But then I would say that that is also happening. That's something you have to, I would say, either we regulate for or we expect it, and you kind of work towards that, I would say. But then, and if you say, then the argument would also like, okay, if I edit me and my child would have this gene or DNA edited, let me tell you how many DNA gets changed within your lifetime. If I go sit out in the sun right now, if I drink alcohol, there's so many external factors that already modify DNA by the time I have my child. I'm very sure this child is very different DNA, like with this one modified that's changed by Cas9. I just want to state those numbers. When my child will not have 100% the same DNA as me, and if I change one nucleotide, definitely there will be more letters within that child. I just want to throw that out there. Yeah, okay, I'm done.
Yeah, I think this is also very interesting, and I think maybe the fear is, and that's also in something that Gwen is saying, that if you would give it to the next generation, so if it would be transgenerational, then it would be maybe dangerous because you create something and you keep it. But I think the CRISPR system is not keeping so much in the next generation because you also lose a lot of this. The chance that you sort of keep it for the next generation is not so high. And yeah, so especially for the epigenetic editing case, you're anyhow not going to keep it because there are 2 rounds of removing epigenetic marks. So the next generation will for sure lose the settled changes, and then there will be a new settlement of the epigenetic potential largely. So the idea that this is such a big thing that you change for evolution and the next generation is going to change is not so realistic.
Thank you all. You all made great arguments, I guess. Had so many questions, but so little time. So I will combine 2 questions from the audience, because indeed there is this fear about for what will be used, and will it be used for the good or the bad? And I can imagine that because of these questions that some things are like stopped by government. For example, GMOs are, I guess in the USA, in some parts, not CRISPR is not used because people are against GMOs. And one of the questions from the audience was like, okay, let's first start with one of the questions, and it was for Pernette and Ven, is your research being hindered by the Dutch or European law?
So I think there is no problem in changing cultured cells, basically. I think the problem comes when you start using embryos. So if you start using live embryos, then you get problems. But of course, we work under regulation of the Dutch government. So we have these regulations to work with GMO. So the things we do are just in cultured cells. So there, yeah, it fits under this regulation. So there's no problem at all.
I think so. To my side, I don't think I don't. We also hold license to work with GMO, but we study mostly fundamental work. And how I do say that there is quite an impact from the European Union regarding GMO is companies. So companies normally have their own R&D research. They were looking into CRISPR-Cas technologies for plants, for example. But when they passed the law, I think it became a bit less. But I think they decided, what I heard as well, they're continuing because they believe this is the future.
Okay, and then someone had a question for Jantine. How can ethicists approach the problem of social fear against, for example, GMOs? Because I can imagine that you as an ethicist can be the bridge maybe between the scientists and the public.
Well, it's a bit a difficult question to answer because so I just gave my opinion, and so my views on this are of course not uncontroversial, and it is within the profession of philosophers and ethicists to, bring very clear positions and arguments. And I do not, like, my views do not represent the views of many of my colleagues. So who should talk with the public? If you would have invited here another ethicist, you might have heard totally different things. And that's, of course, a great good in our society, that we do have these debates, for example, on the use of embryos, and even on specifically making human embryos, not with no intention to use them for a pregnancy, but to be able to use them for research. On that topic is right in this moment a big, say, public debate in the Netherlands being started, and the outcome of that is still open. Of course, now everybody is busy to survive like a virus, where we hopefully can use our technologies to preserve life that's currently already there. But at least last year, before this outbreak, the top of the agenda was, one of the agendas in the Netherlands specifically was this: whether, what the law should allow and not allow with human embryos, and whether we should change regulations. And I wanted to ask that, like, also to population, because it would be a change of the law. And I'm absolutely sure that in events there would be ethicists and people from religious studies, political people would bring completely contrary opinions. So it's very difficult. Ethicists will never be able to influence the opinion of the public in one way. It will hopefully make the differences bigger. And with GMO, you mentioned the US. So in the US, genetic modified organisms, that usually plants, for example, in food, are allowed. So on my Kellogg's cornflakes package here, and on my Milano double chocolate cookies, there is, like, it says how many calories. So don't eat too much of it. But it also says part of this project has been made with genetically modified ingredients. So I don't care. But I see in the supermarkets, people like looking at the package, and then they go for another brand. So I actually think, honestly, that may be the way it works out in, say, free societies, that, like, people have the choice. Of course, and maybe this to the embryos and the whatever babies, nobody should, of course, be forced to make a choice in one or the other direction. It should be, and that is also with the cosmetic surgery, nobody should be under an obligation, and nobody is, to have breast implants or an augmentation or whatever big bumps from which they die and so on. If it's your choice to do that, then, and you pay for it yourself. Unfortunately, we cannot stop it, because there are also victims there, as these things may go wrong. But if the options are, if a society is rich enough that can be the point they worry about, like how big your lips are, I think if that would be our only problem, well, then ethicists would be completely redundant, because if there's nothing more serious to discuss, then we would be very lucky.
Thank you all so much for all your great answers. Regarding the time, I think it's best to continue to our last part of the evening, the role of women in STEM, because we're also organizing this from out the WIF students, and we serve the interest of female STEM students at the UvA. Because Emanuelle, Charpentier, and Jennifer Doudna are the 6th and 7th women who won a Nobel Prize in chemistry since 1901. Which is more than 100 years. So 4 out of the 7 women were awarded a Nobel Prize in chemistry in the last 11 years. So do you think that there's a shift in the appreciation of the contribution of women in STEM? And besides that, I will also want to ask you, because you're all doing great in your fields and you have all different kinds of positions when you're doing a PhD, Jantien, you're assistant professor, and Bernette, you're associate professor. And from the numbers of the University of Amsterdam, we can see that the higher the academic position, the lower the percentage of women. So professors, they're only 8% do the science work. So from your own experience, could you tell what your idea is about the distribution of women and men in your discipline?
Yeah, so I've just looked it up in the monitor Landelijk Netwerk Vrouwelijke Hoogleraren. So this is sort of a monitor that in the Netherlands is going on, which is an interesting thing. It started a couple of years ago when the situation was still really in the bad side. But now the numbers are increasing. And so I looked up in the monitor and I noticed that there is 16% of women professors. So this is, of course, the top level then in life sciences. They call it the bio science. So, of course, in the technical sciences, the things are, even worse. So, and then if you compare it with the students, then there is 40% of women students in the life sciences. So this is sort of a really big disbalance, actually, that there are so many students that are women and that there are so few professors. So that is something really weird, and you would consider that could shift easily, but things take time. And some years ago, I was also in the board of the Women of the Faculty Network, so I know a bit of the numbers, and that we are trying to get them really better. So things are, of course, improving, I think, but it's still difficult. And I also know the stories that people started doing sort of the test of when there is a publication, and you do know that that is from a woman or from a man, that it's actually differently judged. Now, I found that really ridiculous, but they have done the test and it seems to be the case. So then if you are on that line, then it's not so weird that also handing out prizes and awards is going into a different direction. Now, I know that at the moment for handing out prestigious prizes also in the Netherlands, that there is always really a good look whether there is a balance in the candidates that are proposed. So I think we are doing better. But yeah, I think this is still something to work on. And I think it's important because the more the picture is out there that, that there is a good representation also by women, the more it will also be seen by the field, and that will really make a difference, I think.
Cool. And Wen and Jantien, do you have something to add maybe?
I guess that it kind of stems off from like the image of scientists back in the days is normally this old white guy with white hair. But that image is changing, right? I mean, that we also see many different people from different countries, and now also more females as well to have these scientific roles. And I think that's great. And also, like I would say, I think I was talking to my colleague the other day, and we say, we think it's hard to become assistant professor, associate professor as young research scientists, because we're women, we're going to be mothers, we're going to have child, and you're going to lose a lot of months and time. And being a professor in academia, it's a passionate job, but it's a hard job. You're going to lose those years that I would say men don't have. They don't lose, and they can push forward, and they will get more papers, and then the whole ball rolls. And I think that's something that people are paying more attention to, that it is okay, if I don't publish 3 papers in a month because I just got a child yesterday or a couple of months ago. And I think that is amazing that and this idea kind of shows that women are capable to do amazing science, right? We're not here to do a hobby, like we're good too.
No, that's really true. Thank you for that. And Jantien?
So in my field, I tried to, and I could not find any source with numbers to look it up. And of course, in science, we prefer to talk about facts and numbers. So my very subjective anecdotal perception is that in the Netherlands, we have had already since decades a whole number, a significant number of, say, prominent female ethics professors. That started already in the 80s, 90s. Of course, that doesn't say anything about the numbers, but these women had and have influence. So that's, of course, also important. I think at the same time with the publications, that is in the humanities different. If you have like a really big research article, say in philosophy, that means that you take 2 years to write it. The review is going to take 2 years. If you're totally lucky and are not endless reviews, you will have published it within maybe in 3 years, if at all. If it's then out, who's going to read it? Well, 50 people. So that, for example, makes that the H index for in the humanities, and the H index is often something like whether you can become professor and climb the ladder, is very different than in the sciences. In the Netherlands, in my discipline, I recently checked it for someone. I saw that the H index of the female colleague was 6. Well, in the sciences, of course, you would not become like a full professor for that. It's for us, for me, as a humanities person in science, always difficult to explain because people don't understand that. But I think for both people from the sciences, students, women in the sciences and women in the humanities, what I really would say, also from the place where I am now, and that has not been easy road, people who know me know that. So don't give up. I got a Marie Curie Fellowship from the European Commission, and I really want to acknowledge it because I'm absolutely grateful that I got that opportunity to go also to Harvard on behalf of the European people. And that was a dream for me, of course, because Marie Curie has been my big hero since I, as a child, read a book about her. So getting much later in life, a Marie Curie Fellowship to promote my academic career, that was really a dream come true. And I think that would be, say, my message to like all women also watching, but also to all male persons watching, because ultimately what's the difference? Don't give up on your dreams, like go for it.
Thank you for these inspiring words. Also for the audience, if you're interested in female in STEM, then you can also follow our Instagram page with students at UvA, at Instagram. So thank you for that. Then we also have some people who are eager to have some answers on their questions. So here is someone who asks, is there any risk that crops modified with CRISPR form a danger from non-modified crops, or that the modified crops become a weed? Onkruid?
I think that's actually a very, very good question. So I think that kind of go back nowadays, like if you kind of look at the amount of strains that we have growing now, to be honest, it's actually a lot less. Like this is without gene editing or anything. This is just us as breeders selecting for the best, biggest growing thing that is out there. As far as ecology wise, I think it really depends on the plants. I cannot, if you say, let's say, I would say an example, if you have a plant that grows on very, very little water, needs very little to grow, perfect in very poor condition, and you put it in a condition where there are just normal conditions, like there's enough food and enough water, maybe that plant may outgrow the rest. Yes, but then there would be, I would say there will always be an ecological selection on its own. I think these, so I think it's difficult. It's not like you modify plants, you go like let it out free in the world, But we're talking about genetic modified plants for greenhouses and more, I would say, controlled areas. But I think it's very hard for me to predict anything unless tested in field otherwise.
Okay, thank you. And then we also have a very fundamental basic question. How does the Cas protein find the right spot?
Oh, okay. Okay, I will try. Okay, this is actually great. This is super, super great. Okay. So you have the Cas protein that is guided by the RNA, and it finds these 20 letters, I would say. But apart from the 20 letters that it finds, it has like a prerequisite requirement. So let's just say DNA is made out of GATC. It must first find, I think for Cas9, GG, and then it says, oh, I find GG, and it scans basically all the DNA, GG, yes, and then it says, oh, 20 letters match, oh no, okay, next, GG, yes, oh, 20, oh yes, it matched, and then these 20 letters, they match with the 20 letters on the DNA, and thereby finding the right spot, I would say. But if Bernard wants to add on, that's fine with me.
No, that sounds really a good answer. I think also understandable for how it works. But it's amazing that it works like this. That is, yeah.
Yeah, so speakers, are you ready for more questions, you think? Yeah. Okay, then we have a question for Jantien about free choice. Can we really expect people make an informed decision about these sort of things? Shouldn't authorities take responsibility?
It's a good question. I think authorities always have their own responsibilities, like scientists have their own responsibilities, but I do believe that people are able to make informed choices. There is a lot of effort put in making information available for the broad public. And going back to what was earlier mentioned, like the biohackers and kids. So biohackers are not kids, and kids cannot do that. But what you do see is that increasingly in schools, in the curriculum, these aspects of biology are introduced, and indeed then you have like some very simple things. It's of course in no way like preparing things to inject in yourself. You should never ever inject anything in yourself. Let me be like very clear about that. But it is, the more children are incredibly interested, and you can explain at that level, start explaining to them the mechanisms of how biology works, how genetic transfer from generations works. Think about in Amsterdam, the Nemo Science Museum. Also, many scientists visit classrooms. So yes, I do think the public is able to make decisions, and we should not forget it's really to put these decisions in such an exceptional place. We should not forget that we do make decisions about incredibly important, complicated, and often acute things. And we seem to be able to do that, even that it's simply part of the game, that afterwards you may think, oh, I took the wrong decision. But that is like, that's of course everywhere. Like undergoing complex medical treatment, or even things like choosing a career path, and you picture like the whole line, like what you want to become, and then you put a lot of effort in it, and then ultimately you have overlooked something and you regret it. I still say go for your dreams in terms of a career, but it's also very good to be aware that there is no guarantee that the decisions you make and that they are, say, the best in the long run. So I really would not want to put these decisions about anything genetics or genome editing in such an exceptional place. At this moment, say the general public does not need to decide very much about genome editing, at least in humans. Another point is, and it was the earlier question, things like the GMOs, use, agriculture use, crop use of genetic modification. And yeah, but we will always have also different, there will always be such a plurality of opinions that I do not have particular fears of that going off the rails with harmful consequences for a whole society. But yes, the authorities, of course, the scientists should be very honest. Scientists should also be very honest in saying what is not possible and what risks are, because only based on that very honest information, people can make such decisions. And things like hyping your latest invention, that is really one of the, of course, that is polluting the debate. That's misinformation. We also see it now with the pandemic. That's very, very dangerous, because then you take away I think scientists have a huge responsibility here.
Thank you. And I think this is a great end of this discussion. So it was really a pleasure to have all three of you. Thank you again for being here. For those that are interested in females in science and discussions on that, you can follow the WIF students. Thank you to the audience. The next beta break will be the 11th of February. It's about the sense of belonging in times of crisis. So actually what Geraldine just touched on. This will be during the Face of Science Park event of the Faculty of Science. Everyone is invited. Everyone can come and listen. You just have to register. Thank you all again, and have a good evening.